Hello and welcome to this webinar titled Biopharmaceutical Process Development, Trends, Challenges, Opportunities, hosted by Biopharma Asia magazine and presented by Kumar Gaurav, AGM Regulatory Affairs at Panacea Biotech Limited. This presentation will be followed by Quality by Design, Approach for a Virus Filtration Application, presented by Sudhakar Nagaraj, Principal Scientist, SLS of Global Regulatory and Validation Consulting Group at Pall Biotech. My name is Stephen Edwards, and I'll be your moderator. Before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first presenter. Kumar Gaurav is a seasoned professional with 15+ years of experience, working in various capabilities in drug development and regulatory affairs, functions in vaccines and biologics industry. He has played key leadership roles and strategic planning roles in development, scale-up, technology transfer, devising regulatory strategies, operation and ensuring compliance. He comes with reliable knowledge base encompassing challenges, opportunities, and unique requirements of the industry through various stages of product development through conceptualization, development strategy, to scale up, manufacturing operations, quality compliance, regulatory submissions extending further to commercialization, product registration, and incremental innovation of marketed products, including risk management at each stage. He is a creative problem solver and a team player who thrives in diverse work environments. He exhibits well-developed and demonstrable executive leadership skills and strong backgrounds in product management with an extensive line management experience, along with a proven track record of significant achievements, many of which are now a major part of the company's vaccine portfolio, and several others which are under various stages of preclinical and clinical trials gearing up for market launch. I'll now be handing over to our first presenter. Welcome, Kumar.
Thank you, Stephen. Thank you for a nice introduction. Good morning, good afternoon, and good evening to everyone joining me today. As per your time zone from different part of world, I am Kumar Gaurav, your presenter for the day. As Stephen already introduced my work experience, I have 15+ years of experience in product development, scale-up, technology transfer, and regulatory affairs. Now we are coming down to our topic, the biopharmaceutical process development. There is nothing new in biopharmaceutical development topic, but what new we are talking today are the trends impacting the manufacturing and development space, development approach involving quality by design and process and analytical technologies, and how to commercialize the product with effective scale-up and consulting challenges on the way. Topics we are covering today are current trends and regulation affecting biopharmaceutical development, quality by design, bringing improvement in biological development and manufacturing space, journey from lab scale to commercial, how to overcome scalability design hurdles. What are biologics? Biologicals or biopharmaceuticals? They are simple preparation from living organisms such as drugs, vaccine, antitoxin, monoclonal antibodies, which used as diagnostic, preventive, or therapeutic agents. Examples: MAbs, vaccines, blood components, allergens, somatic cells, gene and cell therapy. How they are different from the small molecules. Small molecule, as you know, as the pharmaceutical products, and biological and biopharmaceutical. Biopharmaceuticals are generally high molecular weight products made with live organisms. So they have their inherent contamination risk involved with that. They have many critical process steps, difficult to characterize. Structure of biopharmaceuticals may or may not be known, unlike small molecules. They are heterogeneous in nature or often immunogenic in nature. If you talk about the biopharmaceutical market, The biological drug market size is increasing year by year. If we forecast from 2024, we are forecasting the biological market size of $394 billion US dollars. It crosses a variety of therapy areas, treating disease such as multiple sclerosis, Crohn's disease, asthma, psoriatic arthritis, cancers, and the multiple diseases which our vaccine products are covering. Number of patents applied for biological products are growing 25% annually. If segment-wise, if we talk about biopharmaceutical market, the monoclonal antibodies are taking the lead, and they are increasing year by year. By 2023, if we forecast the market, the MABs are going to cover 32% of market size, followed by cell therapy and gene therapy and vaccine products. Now talk about the biological manufacturing process or biological development process overview. What a general process looks like for a biological product, be it a cell culture product, viral product, or a bacterial product. There are major components of cell banks. Then we have upstream processes, downstream processes, and the final formulation. If you see on your screen, our process moved from cell bank to shake flask, where we have some media optimization studies, seed bioreactors, production bioreactors, centrifugation, then we have clarification by depth filtration, multiple chromatography steps, viral filtration step, ultrafiltration, and then final formulation. These steps more or less make up your process development part in a combination. If you talk about the process development timeline for biologics, they vary from 8 to 20 years for any innovator product, and some follow-on product from 5 to 8 years. If we talk about different step of proof of concept step, will take about 1 to 3 years of your timelines, followed by your upstream development, downstream development, your characterization of product, bioanalytical process development, formulation, followed by your trials, clinical trials. There are, in process development steps, we have major steps. We have two components, DS, drug substance, DP, drug product. And there are process steps of upstream process, where you have shake flask, working seed, and fermentation, where we have the propagation of the bacteria or virus, followed by downstream process, which is the purification of antigen, where we remove the impurities, we remove the contaminant, and polish our antigen. Formulation, where we add some additives, excipients, stabilizers to increase safety and stability of product, make it more efficient. Final, filling and packaging, which is convenient to use and easy to store it. We can't go to the market with the product or with a vial of vaccine or any biological which have 4-page instruction booklet to open the vial. So it should be convenient to use packaging. Whenever we are developing any process, we have one checklist, how to go ahead with that development process. The checklist contains the DS quality linked to the drug product. We have to check every quality parameter in drug substance before moving to the formulation stage. What kind of process development tools we are using? QbD, process analytical technologies, design of experiments. What should be our development approach? The traditional approach, which are utilizing now, enhanced approach, the QbD-based approach, and a combination approach where QbD plus traditional approach we combine and make it more effective and impactful. The critical quality attributes. What are the critical quality attributes? These are the chemical, biological, or physical attributes that can be defined, measured, and monitored to ensure that we have products whose output remain acceptable as per quality limits. So we have to define those quality attributes before initiating our experiment. Raw material attributes and process parameter to those quality control attributes. What should be our design space? How to design our process, which is easy, scalable, safe, and cost effective. So in process development checklist, we have to check the DS quality links to the drug products, where we have to check all the parameters, all the quality parameters to drug substance before finally formulating it to the drug product. We have to think what kind of process development tool we are utilizing. It is the quality by design, the design of experiment, the process analytical technology, what kind of development approach we are putting on to develop our product. The traditional approach, which we are utilizing right now, the enhanced QbD-based approach, and combination of both QbD-based traditional to optimize our process, make it more effective. The critical quality attributes. Critical quality attributes are the chemical, biological, and physical attributes that can be defined, measured, and continuously monitored to ensure that our product remain acceptable to the quality limits. Raw material attributes and the design space. So which framework our design of product development should fall. The roadmap to our biological process development should have that we have minimum number of unit operation, we have minimum capital investment, we have high cell titers in our product, and we can reduce minimum our operational constraint. So if we go by one by one, then it's process intensification to improve yield, reducing number of unit operations, richer media, feed, and supplements enabling high cell titers, scalable harvest technology that minimize huge capital investments, robust viral clearance approach that streamlines regulatory processes. I'm sure my friend Sudhakar is going to talk about this viral clearance approach in follow-on presentation. Then we have buffer management approach that reduce operational constraint, high capacity chromatography resin with extended lifetimes to reduce the cost. So this is the general roadmap which we utilize, which we use for our biological process development. If we talk about the advancement, the trend which are impacting biological manufacturing right now, what new technologies are coming on the way? These are advanced automation, continuous processing, inline monitoring, and real-time release using process analytical techniques, with eliminating lag between manufacturing and distribution. Plug and play modular and mobile manufacturing systems. Automated facility to make the transition from development to manufacturing seamless. And knowledge management means the documentation, all the back data which make integration across development, manufacturing, and commercial value streams. What kind of challenges we face during biological manufacturing? Any modification in manufacturing process, it impacts identity, quality, purity of the product. Any change in the different expression system from reference drug, there is change in protein translation modification. We have to revise our manufacturing process again. Legal factors, which include patents, trade-related secrets, shorter market lifespan of the product, entry of new products, new biosimilar or biobetters, non-availability of large-scale mammalian culture manufacturing facility. There are different regulations in biologicals, be it the development space or the manufacturing space. Every country have their national regulatory authorities, and US have FDA, EU has their authorities, then we have, India have their NRAs, and the regulation of biological manufacturing, all the different stages, there are different guidelines. One major is ICH guidelines, which impact or affect or which has to be followed through every stage of the biological development process or manufacturing process, be it the cell banking, with the cultural purification steps or drug products. There are different guidelines through every stage of the biological development. Now we talk about the quality by design approach. What is the quality by design approach? It's basically risk-based proactive approach, which involves designing and planning of the drug product or process before actual experimentation of that product. Here we agree on the target product profile, what should be our product, what should be a profile, what will be the quality of that product. We determine the critical quality attributes, which we talked about earlier, which link that attributes to the process parameters. We define our design space, we define our control strategy, what kind of control parameter, what kind of analytical testing we need to do on our product to beat the quality product. Then we prepare our documentation and then life cycle management of that product with continuous improvement. In QbD approach, we can't just make a product and leave it there. It's a continuous improvement. If you see the table here in this slide, you can see that there is difference between traditional versus quality by design approach, QbD approach, in different steps of your product development. In development, the current process development approach is empirical, random, and focus on the optimization of the steps, whereas QbD is systematic, multivariant approach, which focus on control strategy and robustness. In manufacturing part, our current approach is fixed. Once we develop the process, it is fixed. Any change, we have to go through chain controls. We have to take the approval from our regulatory authorities. In QbD approach, there are scope of adjustment adjustable within quality management systems. Then process control technology, there are some in-process testing involved in the current approach. Wherein QbD, it is process analytical technology utilized, process operations tracked and trended along the whole manufacturing step. In product specification, in current approach, primary means of quality control based on the batch data, wherein QbD approach, part of the overall quality control strategy based on desired product performance. In control or testing strategy, in current approach, is generally testing and inspection of the product at the final stage, wherein QbD approach, the risk-based control strategy, in that case, real-time release is possible before final complete testing of the product. Now we come down to the scale up. Why we need scale up and why it is necessary before we go from lab scale to directly manufacturing scale. Scale up is simply an act of designing. Scale up is just the act of taking process from laboratory scale to pilot scale and using that data to design a commercial scale process. It's from 200 microliter or 2 ml to 2000 liter or 20,000 liter. It can be anything. Why scale up? Scale up is necessary. We investigate the product or process in an intermediate scale before putting our money to a full scale production process. Because we can't predict the effect of manifold increase in scale. That's why sometime in biological we go 10-fold scale, 5-fold scale, and then we move forward to the production scale. To gather data which can be used to extrapolate and define the large scale process parameters. A few questions before we plan our scale up. Every manufacturer think about these things like what should be our scale up perspective, why we are doing this scale up, what should be the equipment perspective, what kind of equipment we shall utilize in the scale up process. If you are changing excipient ingredients, anything else that may impact the product performance. What kind of regulatory filing that would be required? If we talk about process scale-up stages, they are basically upstream development, where we check the productivity of the product. Downstream process, where we remove the impurities and check the purity. formulation, and fill and finish development. We check the stability and the storage condition, minimize the stress condition. Common goal of any scale-up is the safety, efficacy, and consistency of the product. In scale-up, we counter the impurities and contaminants. Impurities are internal to the processes. It can be product variants, aggregates, host cell proteins, host cell DNA, buffer additives, media additives, which we have added during the process. Contaminants are basically external to the process. These are viruses, bacteria, endotoxin. These are just some examples. In drug product scale-up, our focus is on formulation, container closure systems, fill and finish process, lyophilization process if applicable to your product, labeling and packaging. Not much of scale-up is involved. Now, how to overcome scalability issues? How is the prior thinking or prior planning? Investing in a scalable process early on can make the difference between advancing through drug development in a productive, stepwise fashion and having to redo early phase work to correct the problem. How to overcome scalability issue? These are some steps I have jotted down here. The early stage biomolecule system screening, assessment of structure stability, post-translation modification, protein sequence variant analysis, which can be performed at preclinical stage during cell line selection, identifying critical quality attributes of the drug substance as well as drug product. Integration of process PAT, process analytical technology, which allow better prediction and control of changes between scales. Single-use equipment and platform solutions, such as fed batch and perfusion bioreactors. Multi-column chromatography, which achieve faster, reproducible scale-up results. Early planning and designing of manufacturing capacity at site scale. When we are designing our process at lab scale, we have to think about the scale-up and manufacturing scale together. What is the strategic approach for successful scale-up? Parameter assessment, cause and effect assessment. What kind of parameters involved in the scale-up and what kind of effect they have on that particular product. We have ranking of process parameters according to their importance. Then we have to assess all the parameters and their impact on the product. Determine the parameter and we have to investigate each parameter individually through experimentation. Then we have to design our product. What is the road to manufacturing? Now manufacturing, what I told earlier, like we have to think about our manufacturing process from early on when we are designing our process, and new technologies, we have to incorporate those technologies to increase our yield, increase our stabilization, process stabilization and quality. So what are those things? Adoption of lean practices by reducing manufacturing operation cost, increase process yield and process stabilization. Agile manufacturing operation with ability to run multiple products in a few lines without compromising quality. Now we have multiple biologics on our way. We can't design single facility for each of them. So we have to have this agile manufacturing operation where we can run multiple products in same lines. Capacity expansion and flexible manufacturing design to suit specific product and market conditions. Expanding manufacturing footprint with strong and capable network with the right supplier and distribution. Digitalization of supply chain to manage inventory, raw material inventory, consumables, distribution logic, and complexity of the cold chain, which is very necessary in case of biologics. What is our decisive journey? This is not only the product development. If we are talking about product development, we have to think about the commercialization of product. Only then we are successful. Only then we have that successful product in our hand. What should be our decisive journey to commercialization? We define product economics from the lab scale based on projected market size and competitive selling, what our competitor is selling. This will guide us for the allowable manufacturing cost. What should be our minimum cost for the manufacturing? Because that alone, after that involves supply chain cost, marketing cost, and conduct laboratory studies and scale-up planning at the same time. Define key rate controlling steps. Conduct preliminary scale-up study to be used in rate controlling step to aid in plant design. Very necessary so that we don't have that much expenditure on plant before we have that scale-up product with us. Design and construct a pilot plant meeting regulatory requirements. Evaluate pilot plans result to make any correction and decision on whether or not to proceed with the full-scale plant or not. What is the road to commercialization now? There are certain recommendations which I have jotted down here. The manufacturing process, invest early in the advanced production process with single-use technology. If you see in the next 5 to 7 years, the major impact, the technology which are going to have major impact on the biological manufacturing is a single-use technology followed by continuous manufacturing. Regulatory processes, establish alignment with the stakeholder, initiate early dialogue with the regulators. IPR, limit your patent litigation. Build out unified patent litigation system within your company for your product. Substitution switching. In biological, still the switching and substitution is very difficult, but start educating physician, patient, and change outlook toward switching and substitution. It's not educating physician and patient, but we have to educate the regulators also. Innovator reach. Differentiate your product with service offering. Use appropriate comparative in cost effective analysis. So this is my part for the product development strategy, trends, and what kind of scale-up issues we can face and how to overcome those issues. With that, I'm closing my presentation with the quote from Greg's thought: "The journey is much easier when you are not carrying your past." True? True for human beings, and not true for biologicals. We have to certainly modify this part. The journey is much easier when you are carrying all of your past for the biological development, all your documentation, all your past experience, because your regulator will ask for everything which you have developed, you have done in your past. So with this, I'm closing my presentation part. If you have any question, please write it down. We will take question after completion of second presentation. With this, now I will hand over session back to our wonderful host, Stephen, who will introduce our second presenter of the day. Stephen, here you go.
Thank you, Kumar. Now, before I introduce our next presenter, Sudhakar, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar, just like Kumar just said. Please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen. And we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Sudhakar Nagaraj has been involved in the pharmaceutical industry for over 17 years and has held positions of increasing responsibility in pharmaceutical QC and QA, R&D, validation, and regulatory affairs. He joined Pall's Scientific Laboratory Services organization in 2006. Currently, he is part of the SLS Global Regulatory and Validation Consulting Team, which is focused on providing streamlined technical guidance and scalable customer-focused solutions to facilitate customer specification, qualification, validation, and regulatory approval using Pall technologies. Examples of customer-focused solutions include application risk assessments for adoption of Pall technologies, including consideration of extractables and leachables, toxology assessments, complex filter validation support, customer-aligned change management, material compatibility information, and QbD/QRM support packages. He is an active PDA India chapter member and represents Pall in Asia. He has completed post-graduation in microbiology and is based in Bangalore, India. I'll now be handing over to our next presenter. Welcome, Sudhakar.
Thanks, Stephen, for the introduction, and good evening to all. So in the second session, I would be presenting on applying quality by design principles for virus filtration. The agenda would include stepwise process for building quality by design principles into a virus filtration process, review how prior knowledge assessments can facilitate the risk assessment that needs to be performed by the end user to determine the most appropriate design of experiment. Considerations for your design of experiment to understand your design space. And discuss some examples of the data that can be provided by the supplier which can be used as prior knowledge assessments. So virus safety, risk evaluation and mitigation is an important consideration in biotechnology manufacturing. So let's look at what processes are at risk of virus contamination. Any biotech process involving use of human or animal derived materials are susceptible for virus contamination. For example, blood or plasma products like factor VIII, 11, immunoglobulins, transferrin, etc., monoclonal antibodies and recombinant proteins, biological medicinal products like heparin, urokinase, hormones, etc., advanced therapy medicinal products, also called as ATMPs, like gene therapy and viral vectors. So these present a new challenge for virus safety. As of now, the guidance documents are under development. It is very critical to understand the potential routes of viral contamination to plan for an appropriate virus clearance strategy. So there are these are the main 4 routes by which viruses can be introduced into a processing line. In case of endogenous viruses, the viral genome is part of the germ line of the species from the cell line. Although these retroviruses do not usually pose a threat to human health, they must be included in the overall risk mitigation strategy. In case of adventitious viruses like BPV, bovine papilloma virus, these are unintentionally introduced through raw materials such as serum, for example. Another possible route may be where a virus is used to transform a cell, and for example it is poliovirus or SV40 transformation. And in case of non-endogenous viruses like parvoviruses, the greatest risk of contamination comes from the cell bank itself. Three main approaches to minimize the risk of virus contamination. So you start with the raw materials required for the bioprocessing application. Possible strategies include using animal origin free materials, screening of master cell banks, implementation of broad screening technologies such as NGS, which is next generation sequencing, which is more widely applied in the industry. And also you look at treating high risk raw materials such as serum using technologies like high temperature short time, which is HTST exposure, which may mitigate the risk of virus contamination. The second step is to have inactivation or removal steps in your process. Minimum of 2 orthogonal steps which works on different principles like low pH and virus filtration by size exclusion can be incorporated in your process. Third is to have product testing, usually conducted at various stages of the bioprocessing application. Testing is typically performed using traditional cell culture infectivity type methods or PCR. However, with both these methods, you need to know what you are looking for. Next generation sequencing is being applied more widely in the industry now. Also, I would suggest to refer this white paper from PPOG, which is released in October 2019, which is about rapid detection of bacteria and viruses in bioprocess sample. And the ultimate goal of this document is the acceptance and implementation of rapid microbiology methods, especially like NGS and qPCR methods for real-time release. Let's look at the overview of regulatory guidelines related to virus safety. These are the documents available for EMA with respect to virus safety, and these are the documents available for US, FDA and USP documents with respect to virus safety. And global regulatory documents are WHO guidelines for plasma-based products and ICH-Q5A for biotechnological products, which is typically followed by most of our industries. Essentially, the scope of these guidance documents can be broken down into design of study, where we would consider number of viruses to be considered, number of test runs, and amount of virus clearance required, and what viruses should be considered for virus clearance testing, like relevant to the process, most resistant, representative, broad range, and how the data is applied to the process, like establishment of log reduction value and assay limits. ICH-QA document is on pharmaceutical development, provides us the definition of quality by design. So quality by design is a systematic approach to development that begins with predetermined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management. Implementation of quality by design into new biopharmaceutical process is now a regulatory expectation, and there are lots of advantages implementing quality by design, like consistent product quality. Implementation of quality by design principles for virus filtration is a stepwise process which starts with defining the quality target product profile, which we call it as QTPP, and virus filtration process. You would consider which critical quality attributes, critical process parameters, and material attributes associated with virus filtration could have an impact. Partnering early with supplier can play a significant role by assisting with prior knowledge assessment and help with the risk assessment of critical process parameters and material attributes. Testing can then be performed to understand the interaction of critical process parameters and material attributes. So this defines what could be the normal operating limits and manufacturing control limits would be. This slide shows an approach that could be used for managing a quality by design program for virus filtration. Determining what prior knowledge exists and building an in-depth risk assessment from this is critical for a successful QBD approach. Once this is completed, filterability studies, which includes protein transmission studies to make sure product yields are appropriate, are conducted. It is appropriate to start thinking of the design of the experiment which will be required for the virus filtration, and for this, bacteriophage studies using the actual process fluid can be immensely helpful to generate baseline data. This can then better define future design of experiment studies prior to performing mammalian virus studies. It is important to notice that the arrows after completion of each step feed back into the risk assessment. The regulatory expectation is that this is a living document which should be updated as more knowledge is gained throughout the life of the process. By following an approach such as this, knowledge, process control, patient safety is increased at each stage while the risk is reduced. Establishing the virus filter design space ICH QA document provides us the definition of design space. Design space is the multidimensional combination and interaction of input variables, for example, material attributes like product pH, protein concentration, ionic strength, and process parameters, for example, throughput, differential pressure, filtration duration, etc., that have been demonstrated to provide assurance of quality. The objective is to fully understand the interaction of product and process parameters to develop a design space within which the operating space such that the filtration is always in control. Many regulatory agencies expect a technical justification for their virus filter selection. Suppliers should be able to provide data to support use of the filter under the intended processing conditions. Virus filter design space is influenced by one, system design, whether it is a batch process or a semi-continuous process or a continuous process, and second, risk tolerance like process interruptions. The virus filter design space can be considered as a cube in which potentially impacting factors are assessed with upper and lower limits established. Within the cube would be the validated filter design space in which the end user can operate. What constitutes prior knowledge? In November 2017, EMA conducted a joint BWP and QWP workshop to understand what actually constitutes prior knowledge and its use in regulatory applications. Prior knowledge could include supplier performance data, published literature, platform knowledge, validation data, established scientific principles, etc. However, data must be shown to be relevant and its use in risk assessment must be justified. So there is a greater expectation for a supplier to be a technical partner to the end user or drug manufacturer. Selection of virus filter. Application of quality by design for virus filter start with selection of a virus filter. Examples of some consideration for virus filter selection are MOC, which is material of construction, compatibility, mode of sterilization, process fluid properties, extractables, desired process parameters, etc. Filter format is also another important consideration. Whether to use cartridge filter format or a capsule filter format in your process stream would depend on the process stream sterilization requirements. Let's look at virus filtration of MAPs. Depending upon the fouling nature of monoclonal antibodies or MAPs during virus filtration can be roughly classified as shown in the schematic. Pall can now provide a filtration solution fitting to all monoclonal antibodies from low to high fouling degree. For low fouling, Pegasus Prime virus filter is typically recommended. So we would suggest to add pre-filter, which is Pegasus Protect, if it becomes a bit more challenging for filtration. For higher fouling monoclonal antibodies, we suggest or recommend Pegasus SP4 with or without pre-filter, which is Protect LV6. Supplier data can facilitate risk assessment by the end users. The data provided by supplier with bacteriophages can help predict virus filter performance with respect to virus clearance and also can help to establish initial design space. However, supplier performance data does not replace the need for end user to perform their own design of experimental studies. It can help to understand what factors may present the highest risk. Parameters which could potentially influence virus retention should be included in your DOE, like process exertions, low pressures, etc. Examples of virus filter design space consideration. These are some of the important critical process parameters which are considered for this study. In case of protein concentration, we use bracketing studies to establish the range where the impact is minimal. For differential pressure, most vendor or supplier can provide data showing the effects and range established where the impact is minimal. Higher percentage flux decay is considered as a worst case. So apart from these other process parameters, regulatory agencies expect to include low pressure and process interruption parameters during validation, since first generation filters, virus filters, had issues of passage with these parameters. Though your actual process may not have any process interruptions, agencies expect to incorporate at least a 10 to 30 minutes process interruption step during the clearance studies. As part of prior knowledge assessment, suppliers provide these data to understand the impact of protein concentration on Pegasus virus filter performance. So the left-hand graph depicts the performance of Pegasus prime virus filter. The study was conducted using 9 gram per liter of MAB. Testing was performed at 30 psi with high throughput and incorporating 30 minutes of process interruption with depressurization at 3000 liter per meter square. And we can see that no MVM was detected in any samples analyzed. And for the final sample, high volume sampling was used after 3000 liter per meter square. The right-hand graph depicts the performance of SV4 virus filter. The study was conducted with 10 grams per liter of IgG, which was quite falling because it was not filtered as well. And testing was performed at 45 psi, and we can see that no PP7 was detected in any samples analyzed, and you can see that it is more than 6 log reduction. Let's look at impact of test pressures on Pegasus virus filter. So this data indicates that Pegasus Prime has a very broad design space in terms of operating pressure, demonstrating over 6 log reduction of PP7 clearance over a test pressure range of 2.5 psi to around 45 psi. Similarly, with the Pegasus SV4 virus filters, you can see over 5 log reduction of PP7 at even the low pressure, which is the worst case, like 10 psi. So impact of process interruption on Pegasus virus filters. So we know that there has been increased interest in assessing the impact of stopping of virus filtration on virus retention. This is in part due to observation that after stopping a virus challenge, the log reduction value has sometimes been shown to be greatly reduced. This effect is largely believed to be due to Brownian motion effect on the filter when the pressure is eliminated, which allows the virus to move deeper into the pores of the filter. When the pressure is restarted, this allows the virus, which has moved deeper into the pores, to travel through the pathway and be recovered downstream of the filter. Therefore, this study was performed to simulate a worst case test with a low flow through the filter followed by a stop in filtration. The pressure was reduced to 0 for 1 hour and then restarted at appropriate pressure. Testing was again continued to 75% flux decay, and challenge fluid was 0.125 gram per liter of unfiltered IgG in phosphate buffer saline. The data clearly indicates that even after the extended stop duration, the filter continued to retain PP7, demonstrating the robustness of this new generation virus filter. On the right hand side graph, the testing was performed using Pegasus SV4 virus filter with 0.1% PSA in at 30 psi. The data demonstrate over 6 logs of PP7 clearance. Pall data can be used by end users considering integrating virus filtration into their continuous bioprocessing application. This study shows that there is no loss in PP7 virus retention when Pegasus Prime is used over extended duration at low flux with long process interruptions of 24 hours. The testing was performed using PPS at a constant flux of around 15 liter per meter square, which is typically seen in continuous bioprocessing application. And we are also developing very similar data with MVM virus instead of a bacteriophage. Here I am presenting the experimental data of Pegasus Prime filters with respect to virus retention regardless of the depressurization cycles. In the first experiment depicted in the left top graph was conducted with 0.125 gram per liter of IgG for 2 hours at a pressure of 30 psi. With a flux decay of 90%, we could see a high titer reduction of pp7 of greater than 7 log. And right top graph depicts incorporation of early depressurization step for 100 minutes and then turned back on, allowed it to go for flux decay, and we did not see any impact on log reduction value. You can see that it is more than, or it is around 7 log reduction. Left bottom graph depicts incorporation of intermediate depressurization step, like we allowed for 40% flux decay and incorporated 100 minutes depressurization step and allowed it to run for 90% flux decay, and again we did not see any impact on log reduction value. We can see it is still more than 7 log reduction. Right bottom graph depicts incorporation of 5 20 minutes depressurization cycle, starting at 90% flux decay, 70% flux decay, 50% flux decay, so that I can incorporate 5 depressurization cycle, and again we did not see any impact on log reduction value. It is more than 7 logs. So performance of virus filter scalability. The experiment done over a small scale filter should be scalable and representative of large scale process filters. So this experiment was conducted to demonstrate scalability of Pegasus Prime NP1 filter, which is a small capsule filter. The virus filter was filtered with 200 liter of PBS, spiked with pp7, and was collected in 4 50 liter liquids. Then there was 5-hour depressurization or pressure interruption step. Further, the filter was flushed with 10 liter of water. The data showed more than 5.6 log reduction that matched with the floor scale study. So use of single-use system becomes very handy to carry out these big experiments to handle huge quantity of product and also spikes. This is a case study from an end user. In terms of flux decay, the blue line represents the prior knowledge assessment, and that's Pall data generated with Pegasus Prime virus filters, spiking with pp7. There was no loss in titer reduction up to 90% flux decay with pp7. And we have end user data from 3% flux decay up to 90% flux decay with MVM and Rio 3 viruses, and Pegasus Prime retained both. So no viruses was detected in any samples. So end user design space under their conditions, Pegasus Prime retained viruses as predicted. This is another end user case study. End user use of Pegasus Prime over a broad operating pressure range and volume throughputs. In terms of operating differential pressure and volume throughput, Pall data indicated as blue line are throughputs from 50 liter per meter square to 3750 liter per meter square, and data on operating differential pressure from 1 psi to 45 psi indicated that no recovery of MVM was measured. And we have end user data indicated as green lines having operating differential pressure from 10 psi to 35 psi and volumetric throughput from 1000 liter per meter square to 3000 liter per meter square retained MVM, and no viruses was detected in any sample. So customer design space confirms the robustness of Pegasus Prime filters in retaining viruses. So this slide is a compilation of Pall data and also from more than 50 customer virus validation data. It is a confidential data, but we have taken permission to compile and present the data. The Pegasus Prime filters are qualified over a very broad range as indicated with blue lines, and all customer data, which is indicated as green line, shows the validated design space. The Pegasus Prime filters are very robust and 100% showed no quantifiable recovery of viruses like MVM, SV40, PPV, CPV, MULV, and Rio3 viruses. So, in summary, implementation of quality by design for virus filtration requires a solid understanding of virus filter design space, and we should consider interaction of critical process parameters and material attributes. There is an increased recognition of prior knowledge. It should be of good quality data and should help with necessary risk assessments. All the case studies we have and also data shared by our customer confirms robustness of Pegasus Prime virus filter design space. So this brings to the end of my presentation. Thank you very much for your attention, and I'm open to have any questions, if any.
Thank you, Sudhakar, for that presentation. Now, we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is for Kumar, and our first question will be, what challenges do you foresee in single-use systems?
Hi, thank you, Stefan. The challenges I see in the single-use system are basically the lack of standardization as of now, the limited scale capabilities. The single-use system are not for huge scale right now in a present structure. The robustness of single-use system, the equipment we are using in single-use system, the bags, they are still not that robust. And issue of extractable and leachables.
Thank you. Our next question is, what is innovation priority area for the companies in biopharmaceutical manufacturing space? Again, the question is for Kumar.
Okay, the priority area for biopharmaceutical industry, if we look around what manufacturers are developing right now, the biosimilars, biopharmaceuticals, so the first and foremost priority area is the bioanalytical capabilities. Then the cell line development and optimization of those cell lines. These are the major two development or priority area right now for the manufacturers, followed by the downstream process, upstream process, and the last but not the least, the aseptic technology and finish operations. These are the current 5 major priority area for the manufacturer right now in the biopharmaceutical space.
Thank you. Moving on, our next question is for Sudhakar. And our next question is, at what phase should we look at virus clearance studies for biosimilar manufacturing?
Yeah, good question, Stephen. So typically for a biosimilar or a generic drug, we should look at doing these studies, virus clearance studies during phase 3 using 3 to 5 viruses. And this is the same expectation as both Europe and US as well. Yeah.
Thank you. Our next question is for Kumar, and it is, what will the top 3 commercially viable biopharmaceutical products in the next 5 to 7 years?
Thank you, Stephen. If we talk about next 5 to 7 or 5 to 10 years down the line, I have already presented in my slide that the major products which we are seeing which are commercially viable are MAB, monoclonal antibodies, cell and gene therapy. As you see, lots of new gene therapies are coming up, and the vaccines, they are still in the market and they are going to be. Thank you.
Thank you. Moving on, our next question is, can Pall help in addressing some of the issues related to filtration during virus clearance studies? That'll be for Sudhakar.
Yeah, thanks, Stephen. Yes, definitely Pall will partner with the supplier or the vendor who actually performed the virus clearance study along with the drug manufacturer. And we all three should work together to ensure the virus clearance studies are successful. There are many pitfalls which, with our experience, yes, we can help the drug manufacturer in getting those pitfalls cleared.
Thank you. And our next question is, can we use only one virus, MVM, during virus clearance studies, and is it acceptable for regulatory agencies? And again, that will be for Sudhakar.
Yeah, EMA has very clearly stated that yes, we can use only MVM, but before we assume that this is applicable to all the products, we suggest or we recommend that we have an initial dialogue with the regulatory agencies before we carry out the test. So as of now, yes, EMA is okay by conducting virus clearance study with only MVM.
Thank you. And with that, we will conclude the question and answer session. But I do believe we actually have a question for our audience today, and that is, if you do have need for process development services and would like Paul to contact you, please use the buttons below under Vote from the Audience to click yes, and we'll get in contact with you. Before I finish the webinar, I would like to ask our presenters, Kumar and Sudhakar, if you have any closing remarks. So Kumar, do you have any closing remarks for our audience today?
Yeah, thank you. Thank you, Stephen, for nicely conducting this webinar. And thank you, everyone, for your patience. We had small hiccup in audio, but I hope presentation enriched you with new development in manufacturing and development space. If you have any question, you can write me. My contact details are given in end slide of my presentation. Thank you once again. And thank you, the team Biopharma. Thank you.
And Sudhakar, do you have any closing remarks for our audience today?
Yeah, thanks, Stephen, for giving us an opportunity to present in Biopharmaceutical. And I encourage audience to ask questions or write down the questions to us, and we will definitely revert back with the answers. And Paul is willing to partner with the customers in executing the virus clearance studies as well. Thank you, Stephen.
Thank you. And I would like to take this opportunity to thank our presenters. Once again, that was Kumar Gaurav, AGM or Regulatory Affairs at Panacea Biotech Limited, and Sudhakar Nagaraj, Principal Scientist of SLS Global Regulatory and Validation Consulting Group at Pall Biotech. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.